Bismillahirrahmanirrahim. Today I am going to discuss about de novo synthesis of fatty acid. It means de novo synthesis means newly synthesized within living body. So de novo synthesis of fatty acid, it means synthesis of new fatty acid within living body. Now, de novo synthesis of fatty acid starts from glycolysis. When fatty acid is synthesized and deposited in our body, it's a question for Viva. Answer is in oil-fed state, or oil-feeding state, when we take adequate glu- glucose or carbohydrate during feeding. So when glucose level is more, glycolysis run fast, and ultimately acetyl CoA, oxaloacetate, acetyl CoA, and cited synthesis occur. During more glycolysis, we can get more cited. Ultimately, more TCA cycle. More glycolysis, more TCA cycle run occur during more glycolysis. When glucose level or glucose intake is more. So after taking more and more carbohydrate, we become fatty. Later I'll show you how we get fatty taking more and more carbohydrate. Now, more and more glucose, more and more glycolysis, more and more TCA cycle. So ultimately, cited synthesis more occur. Now, you follow here. This is the mitochondria, outer and inner layer. This mitochondria is not permeable to acetyl CoA, but it is permeable to citate. So, this citate is permeable to mitochondrial membrane. So there is a cited shuttle mechanism. Through the cited shuttle mechanism, cited come out the mitochondria. Here you follow cited come out the mitochondria through the cited shuttle mechanism. This acetyl CoA is synthesized within mitochondria, but it is impermeable to mitochondrial membrane. That's why acetyl CoA with oxaloacetate is converted into cited, and through the cited, it ultimately acetyl CoA come out. It means cited come out the mitochondria. Here follow cited is now converted into oxaloacetate and acetyl CoA outside the mitochondria. This is the cytosol. Cited come out the mitochondria. We know cited is 6 carbon containing substance. Now cited is break down into oxaloacetate, 4 carbon containing oxaloacetate, and 2 carbon containing acetyl CoA. This acetyl CoA needed for synthesis of fatty acid. You follow here. 4 carbon containing oxaloacetate is converted into malate by malate dehydrogenase. It is a reversible reaction. Now, malate is converted into pyruvate by malic enzyme. Here, a reduced coenzyme NADP is converted into NADPH2. This step is very much important. Malate is converted into pyruvate by losing one carbon as carbon dioxide. Here follow here malate 4 carbon, pyruvate is 3 carbon. Here 1 carbon is lost as carbon dioxide by the help of malic enzyme. And here NADP is converted into NADPH2. And we can get NADPH2 through the SMP shunt. During glycolysis we can, or glucose oxidation, we can get NADPH2 also through the SMP shunt. Hexose monophosphate shunt, through the hexose monophosphate shunt pathway we can get NADPH2 from glucose oxidation, and we can get NADPH2 from malate to pyruvate. This is step. NADP is converted into NADPH2. This NADPH2 also important for synthesis of fatty acid. Later, I will show you how NADPH2 helps in fatty acid synthesis, how acetyl CoA helps in fatty acid synthesis.